Hi everyone, today we'll be talking about proteins and specifically we'll be going over different types of amino acids, different components of amino acids including side chains, as well as some important components like peptide bonds and important key terms to know that relate to protein conformations. So we're going to start by going over amino acids, which is the building block of a protein. So these are the smallest units of proteins, and they're composed of alpha carbons, amino groups, hydrogens, carboxylic acids, and an R group, which can vary between amino acids. So here I have a generic structure of an amino acid, and we're going to go through and quickly identify which components are being referred to here. So here we have the alpha carbon. So I'm going to draw a line to this one. The amino group refers to the N bonded to the two hydrogens. The hydrogen is, of course, the hydrogen here. The carboxylic acid is the C double bonded to the O and the alcohol. And the last thing is this R group here, which will be different depending on the amino acid that you're looking at. So we're going to go over the different types of amino acid side chains that exist, and the side chain is also the same thing as the R group, so this is the thing that varies across all the amino acids. And for our purposes today, it's not as important for you to know how to draw the amino acids or even to memorize the structures of each individual one. That's something you would be more likely to do in an introductory biochemistry course. But for organic structures and molecules, it's important to know which amino acids fall into which categories, as well as what components and aspects of the side chains make them fall into these categories. So we have hydrophobic side chains and hydrophilic side chains. And within these categories, we have hydrophobic non-acidic side chains and hydrophobic acidic side chains. And similarly, we have hydrophilic non-acidic, acidic, and basic side chains. So we're going to take glycine, for example, and we're going to try to see and understand why is it considered a hydrophobic non-acidic molecule with its side chain. So here's our alpha carbon. We have that hydrogen coming off of it. And the interesting thing about glycine is that its R group or side chain is also a hydrogen here. So obviously hydrogen is considered hydrophobic, and it's non-acidic because it's not able to protonate another molecule. So this is not a proton donor, and so it's considered non-acidic, and it has to do with the pKa value and such. And if you want more information about acidity and basicity, I have a video on acids and bases in organic chemistry, and a lot more about how proton donation and proton acceptance works. So for more context, you can refer to that video, but it's a particularly important concept to know for amino acids. Here we have alanine, and here's our alpha carbon. We have that hydrogen coming off of it, and here is our methyl group, which is our side chain or R group here. And methyls, of course, are hydrophobic as well, so they don't interact well with water at all. And this can't release its hydrogen to be protonating anything else, so it's not acidic. Here we have a tyrosine, which falls under the acidic hydrophobic category. So if we were to identify what the alpha carbon is, it's actually this one over here. And this whole thing is the side chain. And the reason this is hydrophobic is that it has a lot of hydrocarbons here. And this kind of negates the polarity of the alcohol that's attached to it. So, and also this resonance here caused by this benzene makes it hard for it to be acidic because it's so stable that it doesn't is not as likely to be able to release its hydrogen here. So not really, or it's so stable that it allows the hydrogen to be released. So this makes it an acidic molecule. Let's look at serine and aspartic acid. So here we have serine, which its side chain is right over here. There's that alpha carbon. So this is hydrophilic, of course. We have that polar oxygen. The hydrocarbon chain's not too long. And it's actually considered non-acidic because its pKa, if you remember, the lower the pKa, the stronger the acid. And this is actually not a very strong acid because it has a pretty high pKa. So compared to something like a carboxylic acid over here. So this makes it actually a non-acidic hydrophilic molecule. Here we have aspartic acid. And aspartic acid, here's our alpha carbon, and here is the side chain. And this is considered hydrophilic, of course. It has these hydrogen bond donors and acceptors. Here's our hydrogen bond acceptor, here's our hydrogen bond donor, and with this hydrogen here that can be released. And this one is considered acidic because it's a carboxylic acid, and carboxylic acids are very easily deprotonated. So this hydrogen can be released into solution, forming a carboxylate ion. Here we have lysine, which is considered a hydrophilic basic side chain. So here we have the alpha carbon, here is the side chain, and this is actually considered basic because this NH2 here can be protonated. If you remember, bases are proton acceptors, so it can actually form something like this, and that makes it as part of the reason why it's considered a basic side chain. So let's look at a chain of amino acids. So here we have a chain of amino acids, and this is something that's referred to as a peptide or a polypeptide, and it's the convention to represent it from the N terminus to the C terminus. So you read it left to right like a book. An N terminus basically means that you start with the nitrogen, or the amino group, and then you end with this carboxylic acid, which has the C for the C terminus, and the N for the N terminus here. So if we were to kind of look at this and identify what are some important components to know, let's take a look first at the amino acid side chains. So I'm just going to draw an arrow to just one of them. There's obviously many. So here is the amino acid side chain of the amino acid. And another really important thing to know is a peptide bond. So a peptide bond is considered an amide, which is basically a C double bonded to an O and to an NH. So it's an amide. It's planar due to conjugation. If you remember, conjugation is caused by 3 overlapping adjacent and parallel p orbitals. So we have 1, 2, 3, this planarity. Actually 4 here. 1, 2, 3, 4, this planarity here. And a trans conformation is favored in peptide bonds because I'm going to move over to this peptide bond here so it's less cluttered. You have this transformation here because it allows more space for these side chains to fit in the molecule. It reduces the torsional strain, and if you could imagine, if this was pointing this direction instead, this whole side chain would be flipped over. It would be kind of fighting for space with this side chain here, and that would be really unstable for the molecule. So a trans conformation is favored. Okay, so let's just take a look really quickly at this chain and see how many amino acids we can find here, because that might be something that you could potentially see. So I'm going to erase some of this so you can see some stuff better. Just do that. Okay, that should be good enough. Let's go ahead and see. This is one amino acid. Here's that peptide bond. We have another one here, another one here. So we have, let's see, 1, 2, 3, 4, 5, 6, 7, 8. So we have 8 amino acids in this side chain, or in this peptide chain here. So the next thing we're going to talk about is protein structure. So there's 4 main protein structures that are important to know. The first is a primary structure, and this refers to the amino acid sequence. So having something like cysteine, lysine, glycine, for example, it's just kind of the chain of amino acids, the simplest form. And here's that N terminus and C terminus again. Secondary structure consists of 3 major motifs: the beta sheet, alpha helix, and random coil. So we're going to look a little bit more at that on the next slide. So secondary structure, and this is when we start having hydrogen bonds between the backbone atoms. And these hydrogen bonds are usually between the peptide linkages in the molecule. So you see here, you can recognize this is our amide linkage here. This is that peptide bond. That's really important to know. So you have that hydrogen bond that's connecting them the two amino acid chains together. Okay, so in a beta sheet, you have these aligned amino acid chains connected by the hydrogen bonds, and beta sheets can be parallel or anti-parallel. So they're considered parallel if the N terminus of each amino acid chain is on the same end, and they're considered anti-parallel if the N terminus are on opposite ends. So if we were to look at this beta sheet example here, here's our N terminus here. For this amino acid chain, here's the N terminus of the other chain. So since they're on opposite ends, this is something that would be considered anti-parallel. The second type of motif is an alpha helix, and this is really distinguishable by this clockwise spiral here. And the side chains point downwards, and here we still have those hydrogen bonds between the backbone atoms. And if you imagine there's like an axis kind of going vertically down, and these side chains or these hydrogen bonds kind of follow the parallel axis of the alpha helix. And then the last thing is a random coil, and this is just a third motif that you should know. There's not really much to say more about it other than it's not really an alpha helix or a beta sheet, kind of folds like randomly, quote unquote, but there's not really any distinguishing feature for it. The next type is a tertiary structure, and this involves the folding of the protein. And now you start to have bonds between the side chains in addition to those bonds between the backbone atoms. So here you can see the side chains interacting together. You have different types of bonds. You start having disulfide linkages for the first time, hydrophobic interactions, which is when you have these hydrophobic side chains interacting with each other. Of course, you have those hydrogen bonds again, and you have some ionic bonds where you have that plus and minus interacting. So you can see how this starts to fold the protein into this structure here. It's looking almost 3D. So in an aqueous environment, the hydrophilic side chains will actually point outward, and this is a really important concept to know because this, the polarity of the side chain affects the folding of the molecule. So when you have an aqueous environment, this means that there's water in it. So hydrophilic side chains will be more likely to interact with this aqueous environment since they can be soluble in a watery environment, and they're, like dissolves like, and they're able to interact together. So these hydrophilic side chains will point outward, versus hydrophobic side chains will actually point inward to stay away from this aqueous environment. They're afraid of the water. They're afraid of the aqueous environment, so they don't want to interact with it. So they'll orient themselves to the inward portion of the molecule. The last thing to know is the quaternary structure. And so now you have all these non-covalent bonds between multiple subunits. And I'm going to have a video also on non-covalent bonds, so you can refer to that if you want to learn a little bit more and get more information about that. But here you have multiple subunits interacting together, non-covalent forces kind of holding these multiple chains together. And this is the biggest and most complex structure of proteins.